Hello everyone, this is Bio 10, Week 3, Lecture 2, and today we'll be discussing about prokaryotes and viruses. If you remember from the previous lecture, we had studied about the domains of life. Everything on the right is made up of eukaryotic cells, the cell type that we studied in the previous lecture. Bacteria and archaea are made up of prokaryotic cells, the primitive kind of cells. So today we will be studying the prokaryotic cells. The learning objectives are structure and function of prokaryotic cells and learning the structure, function and diversity of viruses. When we talk about prokaryotic cells, keep bacteria or bacterial cell in mind. So by definition, a prokaryotic cell is a cell with pro, which is primitive type of karyo, means nucleus, a primitive form of nucleus. That means they do not have a well-defined nucleus, but their genetic material or the nucleic acid is present without a membrane-bound organelle right inside the cytoplasm. Let's look at the eukaryotic DNA. Eukaryotic DNA is present in two forms. One is nucleoid. A nucleoid is a region in the prokaryotic cell which contains most of the genetic information, the DNA that is responsible for providing genetic information to the next generation. It is not enclosed by any membrane such as the nuclear envelope of the eukaryotic cells. DNA is single, unlike several chromosomes of eukaryotic cells. The DNA is circular, it's double-stranded, and is supercoiled to make it very compact. It contains all the genetic information. So the main difference from eukaryotic nucleus is there is no membrane, it is irregularly shaped, and it is a simpler form of DNA, unlike the chromatin or chromosomes of eukaryotic cells. Another place where you find the DNA in a bacterial cell is called the plasmid. So plasmid is a small circular double-stranded DNA which is separate from the chromosomal DNA of a bacterial cell. These plasmids are also replicative and they replicate independent of the chromosomal DNA. They are also inherited to the next generation and they provide antibiotic resistance. The cytoplasm, just like the eukaryotic cytoplasm, mainly contains water, enzymes, nutrients, and waste products, and house several organelles which provide different functions to the cell. These are the site for metabolic activity, glycolysis, protein synthesis, and several other chemical reactions occur in cytoplasm. The cytoplasm has granules. The granules can store different things. For example, a granule may be storing the glycogens, which are the energy source because glycogens are sugars. The granules might also store some kind of phosphates. They also contain sulfur sometimes, and the bacteria can also use sulfur as a source of energy. And sometimes these prokaryotic cells also contain gases inside their granules which provide buoyancy or helps in floating on the surface of water. Just like eukaryotic cells, prokaryotic cells also have ribosomes. Ribosomes are very important organelle. They are involved in synthesis of proteins. Unlike eukaryotic ribosome subunits, they have 30S and 50S subunits, whereas the eukaryotes have 40S and 60S subunit of ribosomes. If you remember, in eukaryotic cells, The ribosome would be either floating inside the cytoplasm or be attached to the endoplasmic reticulum. Because prokaryotic cells do not have endoplasmic reticulum, the ribosomes are usually floating in the cytoplasm. These ribosomes are also a very important target for several antibiotic drugs, because if you can disrupt the protein synthesizing machinery of a cell, you will almost kill the cell. The bacterial cell wall or the prokaryotic cell wall is the rigid structure present outside of plasma membrane in the bacteria, which provides shape and protection to the bacterial cell. Unlike the plant cell wall, which is made up of cellulose, the bacterial cell wall is made up of peptidoglycans. Based on the type of cell wall present, the bacteria can be of two types: gram-positive, the bacteria which can stain positively with the gram stain, or gram-negative, the bacteria which does not take up the gram stain. The gram-positive bacteria have thicker peptidoglycan, like you see here, whereas the peptidoglycan layer is very thin in the gram-negative bacteria. The cell wall is again a very important target for antibiotic drugs, because the cell wall provides protection and shape to the bacterial cell, and if you destroy it, you kill the cell. Now let's have the perspective of how small the bacteria and virus are. Some of you must have seen the egg of frog, which is about 1 mm in diameter. Smaller than that is a human egg, and about 100 to 1/10 is the animal cell or a plant cell. About 1,000th time of this frog egg is a bacteria or mitochondria. So a bacteria is almost the size of a mitochondria. 1/10 the size of bacteria is a flu virus. 1/10 the size of a flu virus is a molecule of protein, and 1/10 the size of molecule of protein is this carbon 60 ring, and you can see the carbon 60 ring is only 10 times bigger than an atom. So the takeaway is that a virus is only 1,000 times bigger than an atom and 1/10 the size of a bacteria. Viruses are microscopic particles which invade the cells and destroy them. Viruses are not cells because they do not have an independent cellular machinery. They don't have their own cell organelles or the machinery for synthesis of biomolecules. Viruses have a genetic material, which can be DNA or RNA, which are enclosed in the protein coat. Viruses are called the obligate intracellular parasites because they cannot grow outside of a host cell. They are very small infectious agents. They cause various diseases in their host body, and they cannot be targeted by the antibiotic drugs. We can target bacterial cells with the antibiotic drugs, but not the viruses. Let's have a look at the basic structure of a virus. The viruses usually have the genetic material inside them, which can be DNA or RNA. They are enclosed by the layer of proteins, a protein coat which is called capsid. The capsid is made up of protein subunits called capsomeres. Thus, the nucleic acid plus capsid makes nucleocapsid. So just this internal structure is the very core of a virus. Then there can be an outer envelope present on the virus, or it may not be present. These envelope particles are derived from the host phospholipid layer of membrane or host fatty acids. Sometimes they also have these glycoproteins called the spike proteins, which are recognized by the immune system, or they are used for recognizing the host cells which the viruses want to attack. Based on the shape of this protein layer, the capsid, viruses can have different morphology. They can be spiral, like here. They can be icosahedral, like here, or they can have a complex structure like we see in case of a bacteriophage. Viruses can also be classified based on the kind of nucleic acid they have. They can have a double-stranded DNA or a single-stranded DNA, a double-stranded RNA or a single-stranded RNA. The example of a double-stranded DNA virus is a herpes simplex virus. A single-stranded DNA virus is a parvovirus. Double-stranded RNA virus is a rotavirus, and single-stranded RNA virus is a hepatitis C virus. Now let's look at how viruses replicate inside a host cell. There are several steps and we will look at them one by one. The first step is the attachment step, where the virus attaches to the surface of the host cell. This process is very specific because there are specific ligands present on the surface of the virus, and there are specific receptors present on the plasma membrane of the host cell. The two interact and make a specific attachment. Then the virus is taken up by the cell through the process of endocytosis, and here you can see that the virus is now inside the host cell. Then the virus breaks open and its contents are released in the host cell. The viral genetic material, which here is an RNA, enters the nucleus, and with the help of viral RNA polymerase, it will start producing the copies of its genetic material. Now the viral mRNA will be used to make the virus proteins, and all this virus needs to make its multiple copies is the copy of its genetic material and the copies of its proteins. Now there are enough viral genetic material and viral protein particles, they will assemble to make a new virus. This virus is then egressed from the host cell. In this process, sometimes the host cell dies and sometimes the host cell does not die. If the host cell does not die, it continues to make more and more virus particles. Viruses can cause several diseases in human beings. Some examples are adenovirus, which causes sore throat, pneumonia, and keratoconjunctivitis. Pox viridivirus, which causes smallpox. Herpes simplex virus, which causes blisters and ulcers. Herpes zoster virus causes shingles. Epstein-Barr virus, which is linked to Burkitt's lymphoma. And adenovirus causes dengue, yellow fever, and chikungunya in human beings. To summarize, in this module we studied the structure and function of eukaryotic cells, animal cells and plant cells, and prokaryotic cells such as bacterial cells, and then the structure, function, and diversity of viruses.